Well, good morning everyone and welcome to the SFAM RUMA webinar. I'm Gwyn Jones, I'm a farmer, chair of RUMA Companion Animal and Equine. I did chair RUMA Farm Animals for a number of years. I also chaired EP RUMA in Brussels, and I've served on a number of organisations and groups in the past, both in the UK and in the EU, and I continue to do so in the UK today. So RUMA Companion Animal and Equine is what we're here to discuss this morning. And first of all, a bit of history. As I said, I was chair of RUMA, dealing with medicines and antimicrobial issues in agriculture from 2013 to 2020. And during that time, at board meetings, the companion animal issue raised its head quite often, and several RUMA members had more to do with companion animals than farm animals, in fact. Such as BVA, NOAA, AMTRA, ARDA, for example. I did resist this at first because I didn't want to lose focus on agriculture, and whilst EP RUMA, the European version of RUMA, started actually a few years after RUMA and instigated by RUMA UK, which I also chaired at the time. As I said, we did have both farm and companion animals within that group, but of course it was doing quite a different job. And at RUMA, during my time as chair, we took considerable responsibility in the UK by accepting Lord O'Neill's 50 milligrams per kilogram target. And not only were we determined to hit that target, but we wanted to do far better, demonstrating that British agriculture is responsible, can take ownership and will deliver without regulation, which is often clumsy, poorly targeted, and sometimes with unintended consequences, as we've seen in other countries. Over the past 5 years, of course, we have now halved the use of antibiotics in UK animals, farm animals, and progress continues under Cat McLaughlin as RUMA chair. Although, of course, it'll slow down as we use less, and several sectors are getting quite close to being as low as they can be. But there is work to do in other sectors, and we still desperately need better data in the ruminant sector, for example, something we're all working very hard on. However, I did promise to look at the companion animal issue and held several meetings with board members who had an interest in this area. And it was decided early on that the new body, should we set one up, would be completely separate from RUMA Farm Animals, with its own structure, its own finances, and separate in every way really, but for the name and any areas where we might have a common interest. I knew a few people in the companion animal sector through my work at DEFRA on the Animal Health and Welfare Board for England, and our Secretary General at RUMA at that time, John Fitzgerald, did attend the Bella Moss Foundation meetings in the past, so we had a connection there as well. And I met with several leading vets and people in the companion animal sector, and Steve Howard's name cropped up a few times, and I inquired, when I was inquiring about a suitable person to assist from the companion animal sector to get this off the ground. Because an organisation, of course, is only as good as the people doing most of the work. And at RUMA Farm Animals, I'd inherited John Fitzgerald when I became chair, who was superb. And then on John's retirement, I recruited Chris Lloyd, who's been doing a sterling job as Secretary General since then. So I met Steve Howard, and I could see that we would get on really well, which is very important, I think, and invited him to attend our early meetings where his knowledge and expertise added a great deal. Stephen did accept the role of Secretary General for the RUMA Companion Animal and Equine Group, and has fitted that work into his busy schedule with his day job at PDSA. We used to meet in those early days at the NOAA offices, if you remember those good old days when we actually met face to face. And I used to call that small group the Enfield group at the time, and we developed the idea as far as we could, and then went back to the RUMA board, who did agree that we should go ahead and form the RUMA Companion Animal and Equine Group, with several caveats, of course, as one would expect. And here we are 3 years later. We now have the RUMA companion animal and equine, and I say equine because Nicola Williams was very influential in making sure equine was included. We have the group. We've recruited a wide range of expert people from the companion animal sector, and Steve will go through those, I'm sure. And we've agreed the use of the RUMA logo. This is very important to me, as RUMA is now a very well-known brand and has a very good reputation amongst many highly regarded people and organisations, such as Dame Sally Davis, Professor Pete Borriello, VMD, and others. And also, you know, past CVO Nigel Gibbons, present CVO Christine Middlemiss, and in fact Christiane Glossop of Wales, Sheila Voas of Scotland, and Robert Huey of Northern Ireland. They've all been incredibly supportive of RUMA over the years. So we put together the RUMA Companion Animal and Equine Alliance group of stakeholders. We have our board and we have our measures and targets group, which is now becoming our scientific group. And that's chaired by Daniela dos Santos from the BVA, who you'll all know. And we envisage the scientific group leading, driving the organisation with the alliance group of stakeholders, discussing, agreeing the proposals, and then cascading that out through the veterinary profession. Whilst the main board, of course, takes care of structure, governance, finance, strategy, and all those things. So there's much to do, and it takes me back to the early days actually with RUMA farm animals, where as we now agree on measures, discuss possibility of targets at national, sector, and even practice level, this world is very different to the one I'm used to. And whilst there are lessons and learnings we can take from RUMA farm animals, we need to recognise that there is little scope for doing the same things in this sector. And as a newcomer with very little expertise, I see the companion animal and equine sector where we have exotics, as well as the ordinary pets such as cats and dogs, never mind rabbits and guinea pigs, and a myriad of other animals. It's more akin to human health than farm animals in the approach needed, to my mind. This is about vets and their clients, or to be more accurate, their clients' owners. The average pet these days is a family member, certainly a dog or a cat is. That means the pressures on vets is immense, and the responsibility and emotions involved is huge. Whilst the farm animal vet will do his or her utmost for the animal in question, there are economic, welfare considerations to be taken into account, and whilst no one is happy to lose an animal, farmers will make that difficult decision. Whereas with pets is very different. Children are often involved, families, no one wants to lose a pet if there's any chance, however slim, of saving it. And the vet is therefore often involved in dealing with the owner or the family, very often more than with the animal itself, which may well have reasonably straightforward issues that the vet could deal with in terms of it technical and medical expertise. And why do I mention all this? Well, as the dog has moved over the years from the kennel to the house, to the bedroom, and to the bed even in some instances, we know from the work done at Bristol University that a family and the dog will share the same bug as when they live together. I did tell you I was no expert. And therefore, to my mind, if there's a perceived risk by some, which is unproven, in eating meat produced on farm, transmitting resistance to consumers, then there is potentially a far greater risk here, I would suggest. Again, you know, I've read of isolated instances of pets transmitting disease to owners and vice versa, which is no surprise if they live together and share a home. Therefore, in the interest of One Health and in the interest of antibiotic resistance, RUMA Companion Animal and Equine has an important job to do. It is, of course, not the only organisation out there, but it has the backing of both the VMD and the CVO in assisting in the companion animal and equine field. And I think assisting is the word here, helping and partnering and assist and helping others. And Steve, of course, will go into detail on the science and give you a far better understanding of this area. But as an outsider, I do see some problems in area for the veterinary profession, which do not arise in agriculture. And another example would be farmers, of course, have been administering medicines and antibiotics to farm animals for a very long time, and under the guidance of the veterinary practice, and they're pretty good at handling animals. But your average cat owner, for example, will that average cat owner succeed in getting Moggy to swallow that pill every day for the next 3 days or more? That's a much bigger challenge, and it's difficult to know whether that is going to actually take place or not. As a farmer, I know that gently putting a pill down a cat's throat is rather more difficult than injecting a cow, a sheep, or a pig. I say that despite the fact that as farmers we have equipment to hand, such as thick gloves and Wellingtons, one of which makes a good restraining device when doing this particular job, I've found in the past. To finish, we now have at RUMA Companion Animals and Equine an agreed set of principles. We have agreed on our structure. We've agreed on measures. We're discussing targets and other things. We have both SAVSnet and VetCompass at our meetings with excellent data, and it's great to have those people working together for the good of the industry. And we agree that trends are probably more important in the outset than too much detail. We're starting with dogs and cats because horses, rabbits, and other things are much more complicated. Therefore, they'll come a little later. We also understand the importance of taking everyone with us. And whilst RUMA Farm Animals' success was the vet and farmer partnership, this is all about vet practices and, of course, large corporations. And we can see advantages there in that when it comes to having standards across all practices under the one corporate body, that could work in our favour. Challenges, of course, are how to set targets, separate species, classification of antibiotics, lowering the use of HPCIAs. I'll refer back to that cat. How to influence behaviour is a big one. We need to carefully shape our message, and when we make any statement or issue guidance, instructions, that needs to be done with great care, and we need to take everybody with us. And all this will take funding, and Steve and I are still working on that. We have a request for some prime pumping funding, which we're waiting for an answer on. And once COVID restrictions are lifted, I'll be looking for funding for the future of this group, and my details are available after today. There are enough people, I think, and organisations who make quite a lot of money out of companion animals and equine, and therefore, rather than ask members to fund this organisation, I'm going to be asking major players to put something back in. After all, this is a very important area of work, and they should be proud to be associated with us as RUMA Companion Animals and Equine, and I look forward to an interesting and exciting future with this group. Thank you very much indeed.
Hello, my name's Steve Howard. I'm Head of Clinical Services at PDSA, but also here today in my capacity as Secretary General of the newly formed RUMA Companion Animal and Equine Group. Gwyn's given you a little bit of background in terms of the development of that group from the initial concept through to where we are today. And you'll have realised that we're still in the relatively early days of our formation. And we don't have all the answers at the moment. But what I do hope to take you through today are some of the key considerations that we'll have to take into account as we develop our strategies and activities going forward. Why companion animals and equine at all, you may ask? We know that in the agriculture sector, over quite a number of years, there's been quite a lot of focus of the potential impact upon the food chain or on the environment relating to the use of medicines in farm and food production animals. And there's been very little focus on companion animals and the equine sectors so far. However, particularly in relation to antimicrobial resistance, we do know that there are 10 million dogs, 10 million cats approximately, and that they do live in very close proximity with their owners. Total antibiotic sales are significantly lower for companion animals, but even low use can result in that resistance to key medicines developing. When 51% of UK adults own a pet, then the numbers do add up quite quickly to be significant amounts. How do we hope to make an impact? We know that, again, in the agricultural sector, herd level decisions can make big differences to usage volumes based on a relatively low number of decision points that impact upon a large number of animals and through influencing relatively few of a veterinarian's customers. However, in the companion animal and the owned equine sector, those decision factors have to be considered by clinicians on an individual case basis every day throughout the day. And quite often decisions are made with a sick animal in front of the clinician and a concerned owner. And that means that the concept has to be forefront of the mind at all times and when dealing with all cases. So what we need to do really is promote a culture where responsible use of antimicrobials is the default approach and the default behaviour in both the consulting room and the operating theatre. To help us achieve this as a group, we've teamed up with some of the key influences within the profession: the Royal College, RCVS Knowledge, BVA, BSAVA, VMD, some representatives from academia and Insight, so SAVSNet and Vet Compass, and a number of those organisations that are able to help deliver change, such as the Major Employers Group and some of the major charities. I haven't named all here, and apologies to those that I haven't named so far. Those are just to give a flavour of the kinds of organisations and groups that we've engaged with so far. One of the first areas that we've started to address as a group is we formed a smaller working party to look at measures and targets associated with antimicrobial resistance or responsible use of antimicrobials in the companion animal and equine sector. There are some measures already published. In the UK VARS report, there are measures based on mg per kg, similar to the agricultural sector usage, and HPCIA usage in dogs and cats. We're also considering whether we should include some indicators of the levels of confirmed resistance that we're seeing in the UK in companion animals and equines. We're looking at whether there are some sector or species level measures that would be useful to inform us of progress. What are the most appropriate species specific measures for horses, dogs, cats, rabbits or others? And we are including exotic animals within the companion animal group. And then we're looking at measures at practice level and recognise that these need to be meaningful. Mg per kg doesn't mean a great deal at practice level, and it doesn't influence behaviours within the consulting room or the operating theatre. So we're looking at practice purchase levels of different products, practice behaviours in prescribing of the different product, particularly those high priority, critically important antibiotics. What percent of consultations result in a prescription or a dispense? What percent of animals that are seen within a practice are receiving a prescription or a dispense of antibiotics? And what's the level of use of culture and sensitivity within a practice or other tools that help to target usage of antimicrobials, such as cytology? All of these are measures that as a group we're considering at the moment, and we have to also ensure that wherever we've developed a measure, that we need to consider the tools and activities that would be required to influence that measure, that would underpin that change in behaviours at practice level. And wherever we have a measure, we should also be aware and risk assess for the potential for that measure to drive undesirable behaviours just to meet the target, and make sure that we mitigate against them. So some of the activity that we undertake as a group will be aimed at clinical behaviours, particularly antibiotic prescribing behaviours, both at practice level and at individual level. The pie chart there shows the SPIVS Fees survey result, which over the years has asked the question around whether antibiotics are given at the time of doing preventive surgery, so neutering of dogs and cats. And where we are at the moment is that you can see we've got 71% in the last survey said that they never give antibiotics at the time of neutering. Another 29% then say sometimes they do, always, or it's left to the vet's discretion. We know from the PDSA POR report that 10.1 million dogs owned in this country and 10.9 million cats. And if 71% of those dogs are neutered, that gives us 7.2 million dogs that are neutered, and 86% of cats turns into 9.4 million cats that are neutered. And even at that 4% level, let's say they always give antibiotics at the time of neutering, that translates into 290,000 dogs, 375,000 cats who have potentially received antimicrobial therapy that is not always necessary. And if you expand that out to the 29%, sometimes always or at vet's discretion, where there is little known control over what is happening at practice level there, then that makes over 3 million dogs or cats. And those figures for always give antibiotics were very much higher than that just a few years ago. So we've got potentially very high levels of animals being exposed to antimicrobials when they don't necessarily always need to be, even just as a result of the preventive surgery that's undertaken in veterinary practice. And that's where potentially protocols and audit come in, and providing they are evidence-based and reflect the view of the profession and key opinion leaders, they can be accepted in practices. The graph that there shows our experience at PDSA. This was 10 years ago, and the red line shows the average of the percentage of preventive surgery cases that were receiving prophylactic antibiosis in our pet hospitals at the time. And that was on a gradually decreasing pathway as the whole concept of antimicrobial stewardship was being picked up by ourselves and the profession. But in September, October 2010, we introduced a protocol that said we will not give antimicrobials at the time of preventive surgery unless there are certain defined circumstances. And as you can see, that led to a very rapid reduction in the provision of prophylactic antibiosis in PDSA pet hospitals to really quite low levels. And that's continued to decline, but it's been on a relatively steady path for the last couple of years, and that range across the hospitals has narrowed down even further. What we also did, we had done a post-operative complication audit prior to introducing the protocol, and we did one following introducing the protocol. And we found that there was no difference in the level of post-op complications that we encountered by introducing that protocol that says we're not going to give antibiotics at the time of surgery. So it can be successful at practice level, and what really helped in implementing that was using the whole team. So the nursing team, particularly in that protocol implementation, were important in just acting as the sense check for veterinary surgeons when they wanted to give some antibiotics. We know there's a wide range of behaviours in terms of culture and sensitivity testing that goes on, and empirical use of antibiotics within the profession. And there's a number of factors that influence that I'll come to a little bit later on. And then we have to consider the vet client and patient relationship. The expectations of companion animal and equine clients, again, have to be managed on that individual case-by-case basis. And as I've already said, often at the time when they've presented their sick pet and they are worried, and they feel that what they need and what their pet needs is to have some medication prescribed. So there's quite a lot of work being done, both in the human and already in the companion animal world, around trying to manage the expectations of clients so that not receiving antibiotics is accepted and doesn't lead to challenge within the consulting room. There's also the challenge of client affordability, and veterinary surgeons having to flex their offering according to what the client can afford. So not all clients can afford the gold standard approach to managing their pet's illness. So we have to be conscious that we need to provide what are the responsible alternatives if the gold standard treatment level cannot be provided or afforded by the owner. And then, of course, we know there are challenges in both client and pet compliance. If the decision is made to give antibiotics, then will the course be completed? Will the dose rates given by the owner be at the stated levels? Will the dose frequency be at the stated levels? We know how difficult it is to get tablets into cats sometimes, so missed doses here and there are, I fear, not uncommon, particularly in the companion animal world. And clients remembering to arrive for the checkups that a vet might feel is important in order to monitor the progress of the condition or the impact of any medication that they've given. And there are also considerations of product or market influences upon veterinary surgeons' ability to use and choose what medications that they use. There's a long-acting cephalosporin injection in cats that has a duration of action of 14 days or more, and many vets are using that partly to reflect the difficulties of getting tablets into cats. It is quite a convenient way of making sure that a cat does get the antibiotic that it needs, particularly. Over recent years, there's been withdrawal of some non-high priority, critically important antibiotics from the market. And unfortunately, that will have had to focus dispensing habits on some of those HPCIAs. And there's been very availability of some of those non-high priority, critically important antibiotics as well, which has left many vets with no option but to use some of the HPCIAs on their shelf. And more recently, there's sometimes a price differential between some of those non-HPCIAs and the HPCIAs, where the non-HPCIAs are actually more expensive for the practice and the owner. And that consideration of affordability can lead to the dispensing of a HPCIA because it's all the client can afford when they are cheaper. And then there's the perceptions of the antimicrobial resistance issue. Again, in the PDSA POR report, we asked the professionals what they felt were the biggest health and welfare issues, if not tackled, within 10 years. And as you can see, antimicrobial resistance is on there, which is great. But in terms of where it ranks, in terms of where the profession see it sitting, it's down there as equal eighth on the chart. And that may well be because it's quite difficult to imagine or visualize what the impact of widespread antimicrobial resistance may well be for both the human population and for our pet patients. or equine patients. So we need to raise that awareness and that level of concern of antimicrobial resistance so that it comes higher up in the ranking of what people see as being a major issue to be concerned about. That doesn't mean that the others aren't important. It can sit there up equal with many of the others. And we also asked what practices do to minimise antimicrobial resistance. And it was great to see that many practices do have protocols in place for staff, but that they also try to manage posters for their clients, or limit the number of antibiotic drugs available to vets. And we need to have a think about the best way that we can both influence veterinary surgeon client behaviour and client expectations and behaviours as we work through this, and provide the right resources or guidance that will do both. So I hope that's given you a little bit of insight into some of the considerations that we're going to have to take into account as we move forward. Thank you for listening. It will be great to hear any thoughts, comments, suggestions, or questions that you have during our panel session. Thank you.
So good morning, everybody, and I'd like to thank the organisers for allowing me to speak to you today in what looks like a really interesting series of webinars. So I'm going to talk to you today about AMR in dogs and horses, and going from the community to the clinic. So in terms of what I'm going to talk about today, I'm mostly going to talk about dogs and horses with some data on cats. And I'm largely going to discuss AMR in the context of E. coli. So I am going to talk about quite a bit of the work that we've been doing at Liverpool over the last 10 or so years. We tend to work on E. coli a lot for our AMR studies because it's found in the feces of most animals, and it gets exposed to antibiotics when it's in the gut, and it's also a pathogen in its own right. So it's quite a nice model, an organism to use to look at AMR. So I'm going to talk about fecal carriage of AMR E. coli. So thinking about the vet visiting dog or horse population, as well as hospitalised animals, which we know have a higher carriage rate. And thinking about feces as well, because we know that fecal carriage is a prerequisite potentially for infection, and that gut can act as a reservoir of AMR bacteria and AMR resistance determinants. So moving on then to clinical infections. So obviously we can look at what the prevalence is of AMR E. coli in the feces, but how does that translate to what we see in terms of AMR causing a problems in our animals as well? And then a little bit about genomic analysis of isolates and how useful that is as a tool really for doing surveillance. So there is no routine surveillance in small companion animals and horses for AMR. However, there is data out there available from the in-house diagnostic labs that many larger practices and hospitals will have, as well as the commercial diagnostic labs. So lots of culture and susceptibility data is available, albeit with certain caveats, which I'll discuss later. There is also limited data on antimicrobial use. And we obviously have the sales data, but in terms of getting more data on how these drugs are being prescribed, projects such as the Small Animal Veterinary Surveillance Network, or SAVSnet at Liverpool, and Vet Compass at the RVC have been mining the electronic health records that are available as part of the practice management software that most practices have, where you can then pull out data in terms of what antibiotics are being used, what species they're being used in, and what conditions they're being used for. So again, there is quite a lot of data out there. So some further considerations: there's obviously a limited range of antibiotics available for use in horses, and new antibiotics are unlikely. And there is obviously concerns about the public health threat if animals are carrying AMR bacteria, so zoonotic potential with owners, veterinary personnel and other animal workers who work in the equine and small animal industries. And then there's infections resulting from resistant organisms, which are increasingly being seen in both horses and dogs. So the potential, for having clinical problems in these animals and actually being able to effectively treat certain types of infections. So if we're thinking about antibiotic use in small companion animals and horses, less than 3% of sales can be directly attributed to these species. So we obviously use a lot less antibiotics in small and large companion animals compared to the livestock sectors, for example. But there's lots of opportunities where companion animals can pick up AMR and also transmit it on. So, 1 in 4 households will own a dog. We'll have very close and frequent contact with our pets within our households. We'll let them sleep in our beds. We'll let them lick our faces. So lots of opportunities for AMR, not just to be transmitted from pets to their owners and their families, but also the other way. The other thing that dogs like doing is eating the feces of other animals, so they can pick up AMR bacteria by eating the feces of livestock, horses, or wildlife, for example. We also keep dogs in other contexts, so there's rescue kennels where dogs may come into those kennels in poor health and require high antibiotic input, and because they're also environments with lots of dogs, again, there's that opportunity for transmission of AMR bacteria if they're present. And then, of course, what we feed our dogs is going to have an influence on what they carry. So raw meat diets, for example, I'm going to show you some data on, can contribute to dogs having a higher fecal carriage rate of AMR E. coli. And then we've got our clinical environment. So we've obviously got a lot of advanced treatments and techniques now we're using on our small companion animals. Animals might be hospitalized for extended periods, and there might be a lot of antibiotic input going into these animals within the hospitals, as well as when the animals have had treatments in first opinion practice and before they then get referred on. So lots of opportunities for transmission in areas where AMR may be selected. In horses, we're obviously used to seeing them in stables or out on fields where they might be kept on fields which have previously been used for grazing livestock, or they might be kept adjacent to livestock. And besides sort of our pet horses, we've also got high performance horses. So show jumping horses or eventing horses, they travel all around the world. Again, they may be exposed to different types of AMR bacteria, and the racehorse industry as well, where they have high veterinary input and might be higher end users of antibiotics than perhaps some sectors of the equine industry. And then again, we've got our hospitals, and this is a picture of the equine hospital at Liverpool, where again, performing lots of complex orthopedic surgery, colic surgery, and there's lots of challenges with trying to keep these environments clean and trying to prevent nosocomial transmission as well. So there's obviously lots of different types of bacteria that we have concerns about with AMR, but our biggest concern, I think, at the moment is the gram-negative bacteria, especially bacteria which are multidrug resistant. And we define multidrug resistance as having resistance to at least one drug in 3 or more drug classes. And a specific mechanism we're especially concerned about in the Enterobacteriaceae family, which is the family which includes E. coli, Klebsiella, Salmonella, for example, are the ESBLs. So the ESBLs are essentially enzymes which can break down drugs from amoxicillin right through to the HBCIAs, the 3rd and 4th generation cephalosporins. And these are highly prevalent in E. coli, which cause nosocomial infections in humans, but have also emerged more recently in community infections in people as well. And whilst there are different types of ESBLs, the CTX family is the one which now predominates. And in people in the UK, we tend to see in E. coli infections 15 predominating, followed by 27 and also 14. And a lot of the time, these genes are encoded on large plasmids, and they're also co-carried with lots of other resistance genes. So quite often when you pull out an ESBL producing E. coli, for example, it's often multi-drug resistant. So another gram-negative bacteria, which is also problematic and causes infections in animals and which they can also carry, are Pseudomonas species. And then another enzyme mechanism which we're also interested in are the AMP-C enzymes. So these have activity against the third generation cephalosporins, but unlike the ESBLs, they are resistant to potentiated amoxicillin. So I'm going to talk quite a bit about ESBLs during. So because we were seeing an increase in reports of multidrug-resistant E. coli infections in small companion animals and horses, we wanted to get some baseline data really on what the fecal prevalence was of AMR E. coli. So we did a cross-sectional survey where we recruited vet practices around the country, and then we asked them to recruit dogs and horses to our study, and we got fecal samples sent in as well as a questionnaire filled in by the owner. And then we isolated the E. coli and looked at what the E. coli were resistant to. So we sampled 580 dogs as part of this survey and 627 horses. And you can see in terms of multidrug resistant E. coli, it was 18% in dogs and higher in horses at 38%. When we looked at things like resistance to potentiated amoxicillin and fluoroquinolone resistance, this was quite low. So less than 10% in both horses and dogs. And then ESBL mediated resistance, this was really low in the dog population, so 0.5% of dogs fecally carried an ESBL producing E. coli. However, in horses, this was slightly higher at 6%. And then in terms of AMP-C mediated resistance, this was quite low in dogs at 5%, and we didn't detect this resistance mechanism in any E. coli in horses. So as I say, we collected a questionnaire from each animal we recruited, and we looked at risk factors for carriage. And some of the main risk factors for carriage were basically for ESBL producing E. coli in dogs was the feeding of raw peltry. So dogs fed raw peltry were 48 times more likely to have ESBL producing E. coli in their feces. And then another more recent study that we did with Vanessa Smith also found the feeding of raw meat diet, basically the dogs were at increased odds of shedding such bacteria. And then for horses, if they were recently hospitalized within 10 days or within 3 months, and then if horses were kept on a yard with a recently hospitalized horse, that was also a risk factor for increased risk of carrying fecally E. coli with the ESBLs. And then the type of yard was also significant. So because the feeding of raw meat diet had come up for a risk factor for carriage of ESBL producing E. coli, we did a small study where we compared dogs on raw meat diets, and we recruited 114 for this, and dogs fed on cooked meat diet, and we recruited 76. And this table 1 just shows the logistic regression model, where it shows whether dogs were at an increased risk of fecal carriage of different types of resistance outcomes, whether they were fed on raw diets compared to non-raw. And you can see across the board for multidrug resistant E. coli, for resistance to the third generation cephalosporins, and any resistant E. coli, that dogs were at increased risk of shedding these bacteria in their feces. And then table 2 just shows some preliminary results from a more recent study that we're doing. So there's a PhD student who's doing a much larger cross-sectional survey on looking at carriage of AMR E. coli in dogs fed on different diets. So you can see so far from the animals we've recruited, and we're up to 200 or so, and the prevalence of ESBL producing E. coli in the dogs on raw meat diets is 23.5%, and the prevalence in dogs in the non-raw diet is 0.9%. So that's quite similar to what we found in our previous cross-sectional study. So this is work that is ongoing. We're also doing work culturing these raw meat diets. They're available commercially as well to look at what bacteria they have in them. So we also did a study to look at the carriage rates in animals which were hospitalized. So we recruited inpatients which were in the hospital for greater than 24 hours across 5 small animal referral hospitals in the northwest of England. And again, we cultured E. coli, looked at what they were resistant to. And there was quite a bit of variation in resistance across the different hospitals. So potentiated amoxicillin resistance varied between 16 to 43%. Ciprofloxacin resistance, so ciprofloxacin being another HPCIA, varied between 8 and 44%. And then ESBL mediated resistance varied between 2, so quite low, up to 34%. And interestingly, when we looked at the type of resistance mechanism that was associated with ESBLs, we found that E. coli with BLAR CTXM15 was identified in 6% of samples. So quite a lot of the same resistance gene that we see causing infections in people. In terms of risk factors, the length of hospitalization was a significant risk factor for the isolation of fluoroquinolone and ESBL producing E. coli, and every resistance outcome was associated with the use of at least one drug. And interestingly, the use of potentiated amoxicillin was associated with the presence of ESBL and AMP-C producing E. coli. And that was because what we found, a lot of the E. coli isolates that we pulled out in this study had both of those resistance mechanisms in the same strain. So you were co-selecting for the other antibiotic resistance mechanism when using potentiated amoxicillin, because essentially those genes were linked on the same genetic element. So we've also looked at the commercial laboratory diagnostic clinical data that is available. And this is some data from dogs and cats from the AST results from Enterobacteriaceae. So this was data analysed over a 2-year period from 2016 to 2018. And the two graphs, A and B, just break the data down in terms of looking at E. coli that was resistant to any antibiotic in blue, which you can see is slightly higher for dogs compared to cats. And then we've got MDR or multi-drug resistance in red, which you can see was around the 15% mark in dogs. And again, just slightly lower in cats with a bit more variation over time. And then we've got resistance to third generation cephalosporins, which are seen in the black line, which basically hovered between about 8 and 10% in dogs and between 5 to 8% in cats. So, this was really useful data in terms of knowing how common ESBL and other resistance phenotypes are in these clinical submissions that we're seeing. And the positive thing was that some of these labs also test for carbapenem resistance, and none of this was detected. And also in SAVSNet, they've also been looking at prescription data, and there doesn't seem to be any indication that carbapenems are being used in small animals, which is good news, because obviously if we're seeing an increase in these gram-negative multi-drug resistant infections, there are concerns about vets moving on to carbapenems to use that drug to treat these infections. And obviously we want to discourage that as much as possible, because carbapenems are really important in human infections, in the treatment of these very multi-drug resistant bacteria that we see in hospitals. In terms of risk factors for E. coli clinical isolates being classed as third or fourth generation cephalosporin, in dogs, referrals only had an increased risk, and whether they had an RCVS specialist on site, and then breeds was significant in some cases. For cats, the sampling site was important, or in fact, oronasal pharyngeal samples had an increased odds, and then if the cat was male, it had a slightly increased odds, and that could be, due to fighting, cat fighting, and basically the different treatments that are administered to male cats as a result of those injuries. So now I'm going to move on to horses, and we did a study on hospitalised horses because that came up as a risk factor for ESBL carriage in the cross-sectional study. So this was a cohort study where we recruited horses on admittance to the hospital and sampled them within 24 hours. And then we sampled their feces every 2 days till they were discharged. And again, we looked at E. coli in their feces, and we looked at resistance to different antibiotics. But for this graph, I want to draw your attention to ESBL producing E. coli. And you can see that there is a really strong increase in the prevalence of ESBL producing E. coli the longer the horse has been in the hospital. So we've got, less than 2% of horses carried an ESBL producing E. coli in their feces on arrival at the hospital, and this goes up to 40% by day 4. So we've done a more recent study, which is a multi-centre hospital study, so to try and see how much those findings basically are generalisable, amongst different referral hospital populations. So this is a study from Kaiser Iskren as part of her PhD. And we had 5 hospitals in this study, including Hospital 1, which is where we did the original study. And you can see again the prevalence does vary quite a bit between hospitals in terms of ESBL carriage in the feces of these horses, going from 9% up to over 90% in one of the hospitals. So higher prevalence of ESBL carriage in horses compared to our small companion animal population, but again a lot of different variation in terms of the prevalence. So we've also done some genomic analysis on the isolates from this study, and we've looked at isolates from which were carried fecally by the horses. We looked at environmental isolates that we took from environmental sampling across the hospitals, and we also collected isolates from them from clinical infections. And the number of resistance genes per isolate ranged quite a bit, from 0 to up to 28, with a mean of 13.8 AMR genes or single nucleotide polymorphisms per isolate. So some of these isolates that we were pulling out of the horses were really very multi-drug resistant. And we could look at the types of genes again that were found in these isolates. So we can see, interestingly, that CTXM15 was present amongst quite a few isolates. And we found this gene in E. coli from 2 of our hospitals, and it was basically found amongst a range of different lineages of E. coli, which is quite interesting. And this is again something new, because CTXM15 haven't really been reported in horses in the past, and we've seen certainly the rise in Hospital 1 from the study done 10 years ago when we didn't find any CTXM15 to this being one of the predominant ESBL genotypes we now find. So we're doing a bit more work on the analysis of the genomes and risk factor analysis to try and elucidate what might be driving that. But the other interesting thing was we found colistin resistance in some of the isolates. So we found MCR9 and MCR8 genes in some of the isolates across 4 out of 5 of our hospitals. And this is interesting because colistin is not used in horses, but polymyxin B is used for treatment of endotoxemia and also SARS. But we don't know whether that can select for these colistin resistant bacteria. But the other thing to consider is, again, going back to the hospital study that I showed you the data on before, if you have, for example, these colistin resistance genes co-carried on the same mobile genetic element as other resistance genes, encoding resistance to commonly used antibiotics in the equine population, you can get co-selection there. And it's really important to understand this because it's obviously got implications for antimicrobial stewardship and how we might want to use antibiotics to prevent this. So also as part of Kaiser's study, we looked at clinical submissions from UK horses across different diagnostic labs. So this was looking more broadly than E. coli, essentially. We looked at other bacterial species as well. So we were able to characterise this data from isolates from their sample site and what they were susceptible to, and whether they were from a referral population as well. And this is just some data from E. coli again, from these clinical infections in horses by sample site. So you can see at the top of the graph, we've got where these samples came from: respiratory, urogenital, skin wounds, surgical site and orthopedic, and then those which are unknown or came in a category of other. So if you can see in the red highlight, we've got resistance to 3rd or 4th generation cephalosporins, and you can see for surgical site infections and unknown, we've got a rate of about 24% of those isolates were resistant to 3rd and 4th generation cephalosporins, and then a lower proportion of resistant isolates amongst the respiratory, urogenital, or the skin and wound isolates. And then if we look at multi-drug resistance, again, we can see that the surgical site and orthopedic and unknowns had a higher prevalence. Up to 50% of those isolates were multi-drug resistant, and then lower for the other sites. So looking at the surgical site is quite important, and we might see different resistance phenotypes and different prevalences of resistance among those isolates. So as I say, we also looked at other types of bacteria as well. And what we saw was there was no readily available treatment for adult horses in the UK in 30% of positive submissions for Enterococcus species, and also for Acinetobacter, 9.2% of positive submissions. So, we are seeing cases where some of these clinical isolates are essentially pan-resistant to the available treatments that are currently available in the UK for horses. And the other thing we saw was an increase in multi-drug-resistant submissions from referral practices compared to first opinion submissions. And again, that's likely to be due to, they'll have different populations of animals, and animals may be more likely to be hospitalised if they're and referral practices. And we know from our study that then they're more likely to carry resistant E. coli in their feces. So just to finish off, we know that antimicrobial therapy is likely to be a driver of colonisation and/or infections of horses and small animals with MDR pathogens. And ongoing surveillance is really important. It allows us to monitor the changing patterns of resistance and also to detect the emergence of new resistance. So in the case of, we've seen the emergence of CTXN15 in some horse populations, and also Pelistin resistance. And that, along with antimicrobial use data, is really important to inform antimicrobial stewardship. Obviously, using the data from the labs has challenges. We're reliant on the lab's interpretation of the data, so they might be using different panels of drugs, and they might be using different cutoffs to define resistance. Submission may be biased, and there might be data missing. And I think it's also really important to distinguish between referral hospital and first opinion submissions, because we know we see more resistance in those referral hospital submissions and more resistance, but we don't really want to drive the use of certain drugs in first opinion practice in case they're not necessary. So being careful in terms of how you interpret these resistance patterns and this resistance data, so that we don't escalate antimicrobial treatment when it's not necessary. And there is definitely a need for harmonisation of susceptibility testing amongst labs in the country and also across Europe. And Enovat is trying to do some of that in terms of coming up with standardised methodology, as well as standardised treatment plans as well. So finally, I'd just like to acknowledge my co-author, Dr. Gina Pinchbeck at Liverpool, and my colleagues at Liverpool and the School of Tropical Medicine, who have all contributed to the work I presented today. I'd like to thank the hospitals and veterinary practices, and also the diagnostic labs that have submitted samples and data as part of the various studies, and also like to thank our funders, and thank you for listening today.
Well, good morning, everyone, and we're now live, and we've come in for the question time, which is always the most interesting. You'll have heard the three presentations, and I've got Professor Nicola Williams from Liverpool with me, and also Steve Howard, of course, from PDSA, and we're looking to answer any questions that you may have. We've got 2 or 3 already, so I think I'll start with those, and then more questions will come in, I'm sure, following the 3 presentations that you've seen. And I see there's a question here from Nikki, wondering if there's certain breeds with health problems that predisposes them to bacterial infections, and could this influence prevalence of AMR bacteria in our pets? So I'll go to Steve with that one and see where we go.
Yeah, I think there's quite a lot of breeds that do have conformational issues that do predispose them to some bacterial infections. Obviously, those dogs with narrow ear canals or folded over ear canals or lip folds, where it's most visible, those areas of skin where the air can't get to it as naturally as it does in other breeds, do tend to act as focus points for bacterial infection. And one of the mainstays in companion animal practice for those kinds of infections is the use of antibiotics. Obviously, the use of cleaning or ear cleaner to try and to clear out the infection and let air get to the infected area is really important. And in our ear protocol at PDSA, we advocate the use of ear cleaners prior to use of antibiotics as well. But yes, I think there's a number of breeds that have those kinds of conformational issues where bacterial infection is more common, and where antibiotics are probably used more frequently than they are in other breeds.
Okay, right, let's move on to a topical subject then. This one's certainly yours, Nicola, I think, from Sally Cutler. Given reductions of antibiotics in livestock, why we're not seeing reductions in raw meat diets over time?
Okay, so I guess as Sally asking why we're not seeing reductions in carriage of AMR in dogs fed on raw meat diets. Well, the current study that we're doing, we're partway through, and we are actually doing a much bigger study. So there will be other risk factors which will contribute to the carriage of AMR bacteria and feces. So we need the full sort of data in really, and to do the analysis to see sort of how much the raw meat diet influence what the dogs are carrying versus, you know, whether they've had prior antibiotic exposure, whether they've been into veterinary hospitals, whether their owners work in veterinary hospitals or human hospitals, for example. But I think certainly from the preliminary data, we are seeing a trend, and that there's still a reasonably high carriage rate of things like ESBL producing E. coli. One thing I would say, that some of the diets that we know are being fed, we can't be sure that the meat going into those diets is necessarily British. Some of it might be imported from other countries. And also, I think there is variation in reductions in antibiotic use across different livestock sectors. So there's a lot of different animal products that do go into raw meat diets. It's not just chicken, pork, it's beef. There's a lot of offal that go into the diets. There's duck, turkey, and also, you know, things like venison and other types of meat as well. So there's a huge variety of different animal products going into these diets. So I think it'll be interesting once we start to look, if we've got enough data to see if there's specific diets that may be associated with the carriage of AMR bacteria in some of these dogs. And also the other aspect of the work we're doing, we are actually examining the diets as well. And I think some people might have seen there was an article in The Guardian over the weekend where there was a report from ECMID in terms of researchers in Portugal looking at some raw meat diets and looking what types of bacteria are in them. And there was certainly the diets they looked at, there was high carriage rates. But again, I don't know what the source of that meat was without looking at the actual abstract. So I hope that partially answers your question in terms of, you know, I think we need more data, certainly for our study, to look at the different risk factors which might contribute to carriage in these animals.
Am I right in thinking that raw meat diets are on the increase? Are they still increasing?
We don't have any real accurate data on this, because we've just done a questionnaire survey where we looked at what people were feeding the dogs, how they were feeding the dogs, and hygiene. So we've got about 50-50 people feeding raw versus those feeding cooked meat diets, and about 2,000 respondents. But people were invited to take part, so I don't think we can really infer that, you know, there's 50% of people feeding raw meat diets. I think it's likely to be much less. I think you can conclude it is increasing in popularity, and perhaps more people are moving towards feeding raw diets, given how many different products there are out there. If you go on the internet and do an internet search, there's so many different commercial products and supplies of raw meat diets now. And then if you go to a lot of city high streets, you'll see there's these shops now which basically are just there to sell raw meat diets. So I think there must be a reasonably large market, and perhaps an increasing market that people are making use of at the moment and capturing. So I'd say it does look like it's increasing, but I'm not sure what the figures are in terms of how much it's increasing compared to people feeding cooked meat diets.
Yeah, thank you. Steve, this is a really popular and topical subject at the moment. Do you have anything to add to that?
No, I don't think so. I think that there's been some really interesting talks just over the last couple of years, and there's a few of those available for free on the internet around some of the considerations of feeding raw meat diets, which does encompass quite a lot of the area that Nicola's talked about as well. We would generally advocate a cooked diet, I suppose, for a lot of the reasons that Nicola has spoken about.
Yes, thank you. And I think this is probably related. What are the key surveillance programs in the UK and perhaps across Europe, from Rennie?
Well, I suppose from an overarching perspective, you might well be better placed to answer that one, Gwyn. But from the perspective of the companion animal sector, I think that was one of the drivers really for forming the RUMA companion animal and equine group, in that there's been lots of good work done over the years in the companion animal and equine sectors on antimicrobial stewardship. The BVA, BSAVA, the Royal College have all done work in this area, including the Royal College building medicine stewardship and antimicrobial stewardship into the Code of Professional Conduct. The BVA and the BSAVA have brought out guidance in this area as well. But what we haven't had necessarily in the companion animal world is that means to bring all that together into one area. And if you haven't got that coordination across the sector, then it's difficult to do surveillance on how we're getting along with antimicrobial stewardship or the levels of antimicrobial resistance that we're seeing in companion animals and equines. So we are hoping that, whilst we don't want the group to be a surveillance group as such, that it provides the framework for us to start to do the measures that I spoke about in my talk, and to be able to report meaningfully on progress we're making in the companion animal and equine sectors.
Yes, I'm sure surveillance will be covered in the agricultural sector of this webinar. Quite a lot going on there. There's a question here from John Redshaw. Two questions actually from John. Is antibiotic resistance the main form of AMR of concern in companion animals and horses? And then to what extent and how does the environment influence the acquisition and spread of AMR in small companion animals and horses? Nicola, do you want to go first on this one?
I can do. For the first question, I guess Steve might want to come in as well on that. So I think certainly in dogs, probably antibiotic resistance is the main issue of concern. I guess in horses, there possibly could be concerns over, worm and treatment and resistance to them as well. But I certainly think antibiotic resistance in small companion animals is the biggest concern. I don't know whether Steve would like to comment on that as well.
I suppose I can only talk from experience, really. I think we know, as I said in my talk, that the antibiotic usage in companion animals and equines makes up only a very small part of antibiotic usage in this country, but there is that element that our companion animals and our horses do live very close to us. We have very close contact with them, and any reduction in antibiotic usage in the right way that protects both the animal's welfare and the veterinary surgeon themselves in making those decisions can only contribute to reduction in antimicrobial resistance. We see it sporadically where it causes a real clinical issue, and quite often once you have identified that you've got a resistant organism in a wound, they can be very, very difficult and very lengthy cases to manage. And the range of therapies that you have available to try and combat that particular organism is limited quite often. So they cause real issues on an individual case basis, and they are of concern in terms of where they sit in the bigger picture as well, I suppose.
Yeah, I guess, certainly from our perspective with our hospitals, we're quite often concerned with MRSP, so methicillin-resistant staph pseudointermedius, which I think is less common in first opinion and more common perhaps in terms of causing problems in referral hospitals. We've certainly had some issues where we've had odd cases of MRSP in animals which are really, really difficult to treat and problematic, like you've stated, Steve.
Yeah.
So, yeah, there are occasions when we do see real clinical issues with trying to treat, but it is, as I guess you say, more on an individual animal basis. We have occasionally had outbreaks of things like MRSA in equine hospitals as well, which have caused a problem in terms of trying to sort of do deep cleans and actually get rid of them out of the hospital and stop them being transmitted. And I guess that comes on to the next question that was asked in terms of how important the environment is, because the environment can mean many things. It can be the natural environment, it's also to the farmed environment, to the hospital environments, or the household environment, really, if we're thinking about transmission and sources of AMR. And certainly the hospital environments, especially referral hospitals, obviously there's concerns over, our cleaning procedures, especially in our own hospital, and the potential for transmission between animals that are put into different stalls, and especially when we have a high turnover of animals in our hospital stalls, similar to the same situation you get in a human hospital in terms of how many people are, basically coming onto wards, etc. So I think the hospital environment, there's a lot of work to do there in terms of infection control and also environmental monitoring, to be able to get an idea if we have got issues around our cleaning protocols, and also do monitoring of what's in the environment, what's causing clinical infections, so that we can get on top of it. And I think there is certainly evidence of transmission within our hospital environments. Within household environments, there's some nice work by Johnson in the States, which shows that E. coli can be transmitted backwards and forwards between our dogs and the household members. So, we're sharing our environment so much with our animals that it's not surprising that things are transmitted both ways. And I think we have to remember, it's not just pets transmitting things to us, it's also us transmitting bacteria back to them. And highlighted more recently with some of the reports as well with COVID, and pets acquiring COVID from owners as well. So, and then the farmed environment, I think there is a little bit of data to suggest that dogs which are either on farms or go onto farmland can perhaps pick up antimicrobial resistance as well, because I think, as I showed the pictures in my talk, of dogs having that lovely habit of eating the feces of other animals. So there is that link. How much it contributes to resistance, it's hard to say, really. I don't think there's that much data, because a lot of the data we tend to have is looking at risk factors and what's reported by owners when we do sort of questionnaire type surveys. So I don't know whether that goes some way for answering your question.
Yeah, I think it's a very comprehensive answer, actually. As a matter of interest, do you employ deep clean protocols in hospitals for farm animals, for companion animals?
We certainly do in our hospitals. We have had a problem with MRSA in our hospital, where we were doing deep cleans and still finding MRSA. And what we found with the MRSA is, because obviously it can be on skin and dust, that we were having to get really high up in the stalls and clean the light fittings and all the ledges and everything else to get rid of it, so we couldn't detect it in particular stalls. And it is quite challenging, especially in our equine hospital, because you've got a lot of fixed pieces, you've got rubber mats and everything else, which are particularly challenging. I think our small animal hospitals perhaps easier to deep clean. And I'll let Steve talk now.
Yes, and part of the design of veterinary hospitals, surgical areas, consulting rooms is considering the ease of cleaning as well. So the kinds of materials you use, even when you're thinking about how you construct a veterinary companion animal practice, has to take into consideration how easy is this to clean, and how might it harbour an infection in the longer term. So we certainly do, but I imagine most veterinary companion animal practices will have cleaning schedules, and we have regular deep cleans scheduled in as well, particularly in the theatre areas and kennels of our buildings. So yes, it should really, I imagine, be part of the routine running of a companion animal practice to do those cleaning routines.
Yeah, because we saw significant improvements in human health, didn't we, when these sort of procedures were introduced a few years ago. There's a question from Sean here. Is the model for antimicrobial resistance transferable to other drug classes, such as companion animal workers?
Okay, I'm sort of trying to pick into sort of what that question is asking. So if you're asking whether resistance is, we say evidence of resistance being transferred to people who are working with companion animals, companion animal workers, I mean, there's certainly data to suggest that vets and people working in veterinary practices have a higher carriage rate from MRSA compared to the general population. And we've done a small study in our hospitals where we looked at fecal carriage rates of ESBL producing E. coli. And there was a suggestion that they had slightly higher carriage rates compared to the data that's available in Europe for carriage amongst the general population. The main issue when you're looking at fecal carriage of resistance in anyone, be it people in the same household or people working in veterinary clinical environments, is trying to get fecal samples from people for these studies. Our study was only small because a lot of the time people don't want to provide fecal samples. So it's difficult to get enough samples to be able to look at that in detail, really. But yeah, there is some evidence to suggest that people working in those environments may have slightly higher carriage rates of AMR compared to people in the general population. But I think we need bigger studies to look again at other factors, other risk factors, which may also contribute to that. And I don't know whether that answers the question that Sean was asking. If that isn't the question you're asking, if you want to elaborate a bit more.
I suppose I would just add, Nicola, that yes, companion animal workers can be a source of carriage, but in the companion animal world, it is quite often the pet owner themselves that has to be considered, as well as the staff within the pet clinics or pet hospitals or veterinary practices. So that transfer of, as you say, Nicola, MRSA is probably the one I've seen most frequently. You never quite know for sure whether it's come from the client or whether it was on the animal themselves.
Thanks, Steve. Right, we're over time, but I'm going to take this last question from Tony. It's well recognised as to which conditions do not require antibiotic treatments in cats and dogs. For example, acute diarrhea. Is it possible to interrogate the data to find out how often such conditions are treated with antibiotics in first opinion practice?
I'll pick that one up initially. We hope so, I suppose, is the answer. We in PDSA, we have got a protocol, an antibiotic protocol that's based around the use of antimicrobials at first presentation, and that mirrors I think it was the FACAVA list of conditions that don't necessarily need antibiotics for initial management anyway, including kennel cough, cystitis, management of cat bite abscesses in many instances as well. So I think Tony's absolutely right that there is a good list there, and practices can build that list into their protocols to say those conditions under which they will or they won't use antibiotics. In terms of the measurement, that we do have SavsNet and Vet Compass who interrogate practice data, and that may well be one of those measures that we can look at and see if it's possible to extract it from the clinical records made by veterinary surgeons. At PDSA, we did an audit which was more of a manual look around use of antibiotics in first presentation of kennel cough. Just to give us an indicator, I suppose, of adherence to that protocol for antibiotics used at first presentation. So even if the clinical databases are not mineable because the data isn't structured in quite the right way to be able to extract it, there are actions that veterinary practices and veterinary surgeons can take just to monitor that use of antibiotics in conditions where it's generally considered that they don't necessarily need to be used in the first instance, if that helps.
Nicola, do you have anything to add to this final question?
Yeah, I was just going to add to me that Steve's mentioned there is data that SAVSNet's looked at in terms of the electronic health record. So there is some data, albeit a few years old, where they've actually looked at prescribing round cases of diarrhea. So there is some data out there, but it is a few years old now. But yeah, SAVSNet, that Compass can certainly access that type of data more widely to look at prescribing around things like diarrhea cases.
Thank you. Can I thank both Steve and Nicola for answering all your questions, and thank you for your questions, searching questions, many of them, and quite challenging. As a non-expert, I'm pleased that I can allow others to answer them. I hope you've enjoyed the 3 presentations and the opportunity to ask live questions, and wish you the rest of a good day, and thank you for your participation.